Good afternoon, everybody. This is Daryl Diamond of the Provider Education Team here at the Association of Community Cancer Centers. I want to thank you all for joining us for today's webinar, The Future of Molecular Testing, Optimizing Efficiency Under Increasing Expectations, as part of the Molecular Testing Peer-to-Peer webinar series. Funding and support for this series is provided by Pfizer Oncology. This webinar will be presented by Dr. Joe Kim and will feature Dr. Lawrence Wagman. Dr. Wagman is the Executive Medical Director of St. Joseph's Hospital of Orange County, the Center for Cancer Prevention and Treatment. He has been a leader in the process improvement initiatives for molecular testing at his center. You can read more about the process improvement work they've done so far in the 2015 January issue of ACCC's Oncology Issues magazine. Dr. Kim is the president of Medical Communications Media. Just a couple quick reminders for today's webinar. If you look at your screen, you're going to notice there's a questions or chat box on the bottom right of the control panel for GoToWebinar. This is where you can ask our presenters questions throughout the presentation. So just simply type in your question in the box and click send, and we're going to collect all the questions and have Dr. Kim and Dr. Wagman answer them during the Q&A portion at the end of the program. We're going to try to get to as many as possible, and it's never too early to start asking them. Also note that there's going to be an email sent to all registrants within the next few business days advising where you can access the archive for this webinar, as well as a link to the slide deck. At this time, I'd like to introduce Dr. Kim.
Great, thank you very much. So thank you all for joining us for this webinar today, and we're going to go ahead and dive right in. This webinar is really designed to talk about some of the projects that ACCC has been involved in, and the one that we're going to highlight today is a 2014 project on molecular testing in the community oncology setting, and this was focused around biomarker testing in advanced non-small cell lung cancer, and it involved learning labs for process improvement. We went out to 8 different ACCC member programs, and this was a collaboration that was supported by Pfizer, and we had every center actually review their testing processes, look at their actual data from their cancer registries or tumor registries, and then after customizing some educational content for each of those centers, we actually spent time conducting intensive learning labs at each of those places. These were interdisciplinary. We looked at their processes. We looked at communication and clinical workflow. And based on that discussion and the output, every center developed a series of improvement plans that were tailored for their own institution. And they used that Plan, Do, Study, Act, or PDSA cycle for improvement and provided follow-up. So sort of with that setting and background, and a lot more information is available on the ACCC website, I'm just going to go ahead and provide a very brief overview of the centers that participated. So here are the 8 centers. You'll see that they are all around the country. And this next slide here shows you that as we've de-identified these 8 centers, we clustered them into 2 categories, those that treat over 100 lung adenocarcinoma patients a year, as well as those who treat less than 100. So as you think about your own program, think about the volume, and you'll notice here that we also highlighted what their EGFR and ALK testing rates were for patients who had advanced stage lung adenocarcinoma. This information, part of it came directly from the tumor registry, and part of it actually required each of the programs to go back into their pathology reports or electronic health records and actually look to see what kind of testing rates, what were their testing rates for EGFR and ALK, because that information, generally speaking, wasn't being captured in the registries. You'll see that the EGFR testing rates, on average, ranged from 50 to 60%, depending on the size and the volume of each of the programs, and then the ALK testing rates were around 47 to 60%. I will say right now that as we had these learning labs and discussions, part of the reason why the rates are what they are included, in some instances, the tissue was not sufficient for testing. In other instances, the testing didn't occur. It wasn't ordered, and we'll get into the reasons why. And there were some other reasons as well that led to these percentages. But at the end, after having discussions once again with each of the cancer care teams, and talking with the pulmonologists, the radiologists, the surgeons, the medical oncologists, and the pathologists, each center once again identified where some of those key opportunities for improvement were and then implemented some plans around it. So today we're going to be talking about this project, some of the lessons learned, as well as Dr. Wagman will be sharing some of the experiences that his program has been involved in and some of their initiatives and projects. But we're also going to recognize that we are living in an era where it's much more of a biomarker-driven landscape in oncology, and how lung cancer screening programs are now bringing even more patients into the mix. And so every program needs to really think about how the whole science and as well as the workflow of biomarker testing will fit. And so we'll be talking and touching upon each of these objectives today. Now, to set some background stage, in 2013, I think many of us know that the College of American Pathologists, or CAP, the International Association for the Study of Lung Cancer, IASLC, and the Association for Molecular Pathology, or AMP, released guidelines. These were the first set of guidelines titled Molecular Testing Guideline for the Selection of Lung Cancer Patients for EGFR and ALK tyrosine kinase inhibitors. We used the basis of this guideline for the fundamental content that we delivered at the Learning Labs, because the guidelines do provide a lot of guidance and instructions and give some evidence-based recommendations as well as some suggestions. And earlier this year, these three organizations announced that, yes, the guidelines from 2013 are good, but they really do need to be updated because there's so much that's changing. And so this press release from January mentioned that they plan to update ALK testing recommendations, EGFR and ALK resistance issues, emerging molecular targets, as well as the need for rebiopsying. So I think we know that this information is coming, and stay tuned. We also know that non-small cell lung cancer is very heterogeneous. And if we look back in history, back in 1987, we were aware of KRAS mutations as one of the driver mutations that causes this cancer, but we also knew that there were a lot of other mutations, we just hadn't discovered them yet. 2004, we knew about KRAS and EGFR. 2013, now there's a whole panel of biomarkers that we've identified, but we still know that there's a significant section of this pie where there are still unknown mutations and driver causes for non-small cell lung cancer. So I think we're living in an era where this is going to continue to evolve and get updated, and we're also living in an age where we will find that newer targeted therapies and approaches will be specifically looking for these biomarkers. So the importance of molecular testing and building that process in an efficient way so that actionable mutations can be identified is going to be really critical to improving outcomes in these patients. So going back to those CAP, IASLC, and AMP guidelines, we know that in Section 1, Question 2, one of the key questions that's asked is when should a patient specimen be tested for EGFR and ALK mutations? And the recommendation has some very clear guidance, advanced stage or stage 4, as well as suggestions on both EGFR and ALK testing, given that there are now FDA-approved therapies for these mutations in this patient population. The recommendation also is that tissue should be prioritized for these two biomarker tests. And so the reason that that is there is that there are a number of centers and programs where there's some discussion and to some extent some dispute, and some oncologists may want to test for more mutations. Others may be a little bit more conservative, and understanding that there are differences in opinion on which biomarkers should we be testing and so on. These recommendations clearly say, well, test for these two because we've got these FDA, and if you're going to test for more for clinical studies or for research purposes, that's to be determined by each institution as well as each treating oncologist. We also know that the NCCN guidelines, which continues to get updated, recommends the testing once again for EGFR and ALK, but they also recommend testing in patients with squamous cell, especially those who don't smoke. And what we'll find in the NCCN guidelines is that they certainly are a proponent of identifying additional biomarkers if it can lead to patients enrolling in clinical studies. So once again, we know that there are a number of guidelines out there that support testing, but it's not quite that clear-cut and simple because there's confusion and controversy. For instance, should molecular testing get delayed until the pathologist really knows the disease stage? Because when that biopsy is performed and the cancer diagnosis is confirmed, in many instances we don't know the stage of the disease. which test should be ordered is still, even though EGFR and ALK are given, should we test for additional biomarkers? That, once again, remains a topic of discussion at many cancer programs. Who should be the ordering physician? Should it be the pathologist? Should it be a medical oncologist, even if one hasn't necessarily seen the patient yet? So these are some workflow issues that need to be identified and resolved if a program is going to develop a really refined and efficient process. And there are hospitals that also deal with a Medicare 14-day rule when an inpatient gets diagnosed with lung cancer, and Medicare often will not cover the biomarker testing that occurs if that test is performed while the patient is still in the hospital. The 14-day rule simply says that the patient must get discharged. 14 days later, you can send that specimen off to a lab for biomarker testing, and then Medicare will reimburse for it. So what should they do? Should they wait? Should they test? So there are a number of these kinds of issues that needs to be, in many cases, individualized per program, but ideally processed in a way so that there are efficiencies. And some of the programs have developed reflexive testing methodologies as well, and we'll get into some of those examples. So just talking about some of the improvement plans, on the tissue handling side, these biopsies are often being performed by either pulmonologists, radiologists, or surgeons, and in some cases they're using a core biopsy needle that cuts a segment of tissue, or in other cases they're using a needle aspiration or fine needle aspiration, FNA, and so they're just really getting cells and possibly some fluid. How do these clinicians performing the biopsies, how can they change their process to obtain more tissue? And what can the pathologists do to help alongside to make sure that there's enough tissue for biomarker testing? And these were some of the discussions that we held during the learning labs, because every center recognized that it really needs to be institution specific. And there may also be ways to reduce unnecessary testing in certain patients. And some centers had already developed these pathology-driven reflexive testing pathways, but they have to refine them, and they have to also make sure that everyone understood what they are and bought into them. So these were some of the examples of the improvement plans that we saw. We also saw examples of partnerships with academic medical centers collaborating with the community hospitals and possibly sharing resources around, for instance, the molecular tumor board kind of model, or looking at other ways where they can identify patients for clinical studies and research. And so these were, I think in every instance, depending on the kinds of relationships community hospitals had with academic centers, these were all at sort of different phases along the pathway. We spent a lot of time talking about health IT because in many organizations, the pathology department may use one electronic platform, the outpatient oncologist may use another one, the inpatient hospital might have a different set. So interoperability issues and challenges, a lot of printing, faxing, uploading, and also health IT really does dive into the question of if a pathology report is first uploaded, but then later gets, it needs to get amended because the original pathology report didn't have the biomarker testing. Does the medical oncologist know that these things are pending? And how does that information get communicated? So at this point, I want to open it up for discussion from Dr. Wagman, hear from his perspective at your hospital at St. Joe's, Dr. Wagman, and if you're on mute, just please remember to unmute yourself. As you think about the different barriers, the journey that your organization has gone through, share with us, how your institution approached this topic of biomarker testing and some of the key changes that you made.
Okay, thank you, Dr. Kim. So we got into this process after the ACCC had done the learning lab. So we were stumbling along for a while, or building things as needs arose. So I think we had a coordinated group of interventional radiologists. They're hospital-based, pathologists who are also hospital-based, and that was very helpful in the initiation of just seeing who was at the table. In terms of an internal structure, we used our cancer committee who is responsible for the quality of cancer care, obviously. At our cancer committee is the American College of Surgeons Commission on Cancer approved. So always needs to have projects and activities. But it makes a real nice platform to bring people together who are naturally already coming together, usually quarterly. So that was one of the structures that we used. We had a process of reflex testing, and I think it probably started many, many years ago. But as changes in clinical cancer genetics have emerged, particularly testing for HNPCC products, if you will, and also for the emerging lung cancer based molecular testing, we used that format of our cancer committee and created an ad hoc reflex testing committee. And essentially what the reflex testing committee did was establish, as we're being faced with all these additional molecular markers, we established the hierarchy for testing. So the highest level for which we had to discuss whether or not to do a test on a certain molecular target was the treatment for that, and how to consider that and create a flow to get that done. The next thing we used was prognosis, and that fell way behind whether or not there was a targetable treatment. So prognosis was more of interest. The last one was research, and the research is really as it's coming along. So there's no direct, unless there's a direct study in place, we're not doing any reflex testing for research that might occur. I think one of the most important features in establishing this is to avoid things that are non-informative. So this kind of blanket testing and all the big panel testing without actually knowing where you're going with the information become very confusing, because the results will be all over the place. It's almost in a way a bait and switch, because you say you're going to test for something, you get back a result, and then you have nothing to be actionable on. So that, I think, is a very important piece. And I'll stop there because I want to make sure I'm answering the question Dr. Kim wants me to.
Yeah, absolutely. I think that as we have gone back and followed up with some of these programs, this idea of establishing either a committee or subcommittee that focuses on either molecular testing or biomarker testing has been a pattern or trend that we've seen in some places. The cancer committee that on average may meet once a quarter often is not quite the place for this kind of established focus group around biomarker testing alone. But, like you mentioned, there's the whole reflexive testing process. And one of the questions that comes up over and over is, how did you get buy-in from all the different specialists and medical oncology groups and clinicians on how that reflexive testing, what it should look like and what should be included? And can you just describe a little bit about that process?
Sure. So when we first put a lot of structure into our cancer center, that was about 7 years ago, we agreed as a group. We have 70 physicians who have signed something called conditions of participation, which means they will act in the cancer center by participating in our cancer conferences, by being part of research, by being available for giving talks and being resident experts. So we had those doctors. As a group, we also adopted the NCCN guidelines as our standard for comparison when we would make recommendations, for instance, or look for direction. So we, through the Cancer Committee, we used the NCCN guidelines and said, we're going to set up this ad hoc reflex testing committee, and we'll go through the entire NCCN guidelines, pick out all the molecular testing that's recommended for treatment, and start there. And then we'll also look at the ones that have prognostic importance and see if we want to do any of those. Our ad hoc committee has a medical oncologist, radiation oncologist, surgical oncologist. I sit on it also as the executive medical director, and a pathologist. So essentially, the key players are represented.
And to get, I guess, buy-in and input from the different, I guess, medical groups, for instance, within medical oncology, if there are some in private practice versus other groups, is there a way for them to get fair representation and input on what's going on?
Yeah, essentially, we have cancer conferences that are either organ or system-specific. So we have a urologic oncology cancer conference, a breast, etc. And at those cancer conferences or at the program meetings that accompany them once a month, the physicians can always say, we ought to be testing XYZ molecular marker. And that would then get passed along to the ad hoc committee for consideration.
Okay. And I know that one of the common barriers that has come up over and over is, on this slide, we talk about tissue handling and even just obtaining adequate tissue. What, in your organization, how have you addressed this topic and this issue of, in the past, perhaps those performing the biopsies just needed to get enough to establish diagnosis, but in today's era of precision medicine and biomarker-driven therapies, they need more tissue. So how has that evolved and changed within your organization as it relates to the different physicians who perform these biopsies?
Yeah, that's a great question, and I don't think that's anywhere near being solved yet. The main critical balance here, there's no physician, let me just back up a little bit. There's no physician who wouldn't want to provide you with an adequate amount of tissue to do all the diagnostic tests necessary to get the patient the best treatment. Everybody's on board for that. But it's like everything else where you have a risk and benefit balance. So the doctors do, say it's the interventional radiologist, they do a quick check with the pathologist that there's adequate tissue for diagnosis. And then within that, and I think you mentioned it already, that you need a certain hierarchy that you have to develop before you would, so that you're sure that the tissue gets processed in an orderly pattern that will give you diagnosis and then treatment critical information, whether that's molecular or histologic. The only way to deal with it is to raise the awareness, because ultimately the doctors are there to generate, the biopsying doctors are there to generate a diagnosis. But sometimes it comes a point either for patient tolerance, so they're starting to have a lot of pain or they're moving around, or the risk becomes excessive and maybe bleeding for liver biopsy or risk of pneumothorax or hemothorax even in lung biopsies, or approximation to critical tissues in the mediastinum that will limit it. So I think the only way to know about it is to really look at your own information and see how successful you've been and what you've been successful with. So if you're successful with 19 gauge needles but not with 22 gauge needles, then somehow that piece of information has to be shared, and a standard operating procedure where 19 gauge needles are used, unless excluded for some reason, instead of 22 gauge needles. But it's a process that has to respect the risk-benefit ratio of doing the biopsy in the first place.
Sure. And I think that's a great point, because as we went around to these different programs around process and around, for instance, like even the size of needles or the types of needles that these various clinicians were using, what we found in many instances, for instance, was that we would go to a hospital and say they had 5 radiologists who performed these CT-guided lung biopsies, and we asked them, well, what is your process? What size needle do you use? How many specimens do you collect? And it varied. It varied even within a single organization. And we asked them, well, would it make sense to try to standardize and develop a standard operating procedure on CT-guided core needle biopsies so that it's not so dependent on who's performing the biopsy, because in some cases a radiologist might say, well, I primarily just use fine needle aspiration, I don't use core needles. And two other radiologists may say, well, I primarily use core needles and I don't use fine needle. And so that was one of those discussions where eventually the leadership and the team of radiologists agreed that, all right, we're just all going to follow this certain process and try to standardize. Have you found that your clinicians have adopted those kinds of, I guess, sort of standardization as well, or what's been the experience and discussions regarding just the process of performing biopsies?
So we started out probably about 6 years ago examining the process for doing liver biopsies, and it wasn't really molecular based. But again, I think I said at the beginning, we had a lot of these pieces in place because we were doing them for other reasons. So when we did that process, we got together with the interventional radiologists, essentially, and the pathologists, and the liver surgeons, and the hepatologists, and we said, okay, these biopsies are not adequate with this gauge needle and this number of passes, and these are. So what do you think? How can we get to where we want to get? And ultimately, we came to a decision amongst us of what would be a standard for liver biopsies. We're essentially in the same process right now of doing a very, very detailed evaluation of the yield and looking not just at the diagnosis, because the liver was just looking at liver diagnosis, but now we're looking at whether or not there was enough tissue available for the molecular markers to be done. So that's the first step of it. And then the second step is whether or not when the tissue was sent out for molecular marker analysis, if you got an answer that was adequate, if that analysis, even though the perception was that you were sending enough tissue, whether or not it actually was enough to get the information you need, taking into account that tumor necrosis and stromal tissue versus tumor tissue. So there are a lot of steps in the process, and it really gets to be the details. And I think the slide you showed with the percentage of tissue that was analyzed is the first step, and then what you really want to know is the percentage of tissue that was analyzed that returned meaningful results to move forward on therapeutic intervention.
Right, exactly. What we found was that in many organizations, the feedback loop wasn't in place, so that the clinician who's performing the biopsy didn't necessarily know what is your percentage of biopsies that are adequate for biomarker testing versus inadequate, and they were just guessing because they didn't necessarily get the kind of feedback that said, yes, this is adequate. Most community-based hospitals don't have a pathologist or a cytotechnician or someone else at the bedside when they're performing the biopsy to know if the specimen is adequate. And even then, it might be adequate for diagnosis, but is it enough for molecular testing? So these are definitely issues that we explored and discussed and looked at ways to overcome. How about the issue of health information technology? I know that health IT is a topic that has come up over and over, and organizations have been looking at things ranging from data analytics solutions to clinical decision support, as well as just efficiencies in overcoming some of these interoperability barriers. Has this whole biomarker testing, has it led to either changes or improvements or any as your team has looked at what kinds of health IT resources you have and how you're using it, how's it made an impact in your organization?
So this is maybe another one of those best laid plans of mice and men scenarios. So our health system went on a single system, health IT system, which one would perceive would be perfect because the radiologists and the pathologists would all be on it. All the doctors practicing at the hospital would have access to the information, so that anything you could tell when the biopsy was done. You could even read the note that the radiologist dictated, so you would know what size needle they used and how successful. And then you could look at the pathology. The disconnect comes when the molecular testing is done outside of the institution. And then that information has to get back into the medical record with the pathology report that has the diagnosis for the clinician to then access to make decisions. And in the particular system that we had, there was no notification when the new information came in. So we had to go back to the basics, essentially, of a telephone call or using some of the email or electronic-based communication platforms to say new information has been added to your patient's pathology report. So it
sounds like, I mean, it's every time organizations look for ways of finding and overcoming certain efficiencies, it's often that, it then leads to other problems or scenarios that they have to now address, either through patches or, workarounds, so to speak. I know that the biomarker testing done at outside labs has been a common theme because in many cases you may send your sample off to your standard reference lab, but occasionally the medical oncologist may insist on either a broader panel of tests or additional tests. You may have to then send it off somewhere else. And I know that another issue that has come up over and over is that when that preliminary path report is being reviewed by the medical oncologist, in some cases that preliminary pathology report may not indicate whether or not the sample has been sent off for biomarker testing. And so the medical oncologist doesn't even know, well, should I be expecting EGFR now test results? Do I need to order it? And so some organizations insisted that, all right, well, even that original path report should indicate at the top that either a specimen has been sent off for biomarker testing or testing was not performed for reasons X, Y, and Z, or, here are the test results, even in that preliminary report, so that whoever pulls it up and looks at it has that information. And we've also found in some cases where, the preliminary path report might be dated with one date, but then when the biomarker test result comes back, say, 10 days later, now you've got this additional report with a different date, and it became a bit of a challenge for medical oncologists who were trying to find that information. So they had to find workarounds, especially if they were scanning and uploading documents. So these seem to be common barriers and issues that a number of organizations continue to struggle with, even in the 21st century. I do want to remind everyone on the line that if you all have questions, please type them in. We'd love to address some of the questions that you all have, just to kind of move us through the webinar and to get to some of these last points. We certainly are living in an era where the future direction, as we think about the key trends of this whole biomarker-driven medicine, precision medicine, evolving guidelines, even the biomarker testing guidelines. We anticipate new guidelines probably in 2016. But at the same time, there's this growing importance of how do we deal with patients who have rare driver mutations? How do we identify them, and how do we adequately find these mutations? The clinical research, and I will be talking with you some more about this, Dr. Wagman, because I think in the community, in general, clinical research or patients being enrolled into clinical research, the numbers are often in the small, lower single digits. But in your organization, I know that you all have made some leaps and bounds in advancing clinical trial and sort of research enrollment. Can you just share a little bit about what's been happening there and how you've achieved that?
The primary way of achieving that is making that a part of all our cancer conferences. For every time a patient's discussed, we have our research portfolio. We put it up on the screen so the doctors and nurses and other members of the cancer conference, which is the equivalent of a tumor board, I should say that, see what's available. We're also have a relationship with the medical oncologists where we get either daily or biweekly reports on new patients they're seeing. One challenge for us, and really this is one that we keep trying to think about a solution for, is not so much the newly diagnosed patients, but particularly germane to our conversation today, is the patients while they're recurring. And I think that's another large target area for potential research patients, and it is harder to identify those because they're generally rushing through the offices fairly quickly, and they get on to treatment, and they might not be as easily identified. So that's one way, is just to be there and getting that information. The cancer conferences where the presentations are done, and then we are trying to do some of the really forward-thinking types of clinical trials that use molecular-based targeting and a drug to match the target. And I know that the NCI has just launched their version of it. There are some in industry also. And so that helps keep everybody aware of clinical trials, and it gives them something exciting because now there's a trial for the second line or the third line patient.
Sure, and I think that we're going to be hearing more about that. I know that ASCO also recently announced an initiative around that sort of same approach. One of the questions that's come up, and it's more of a future sort of looking question, is there's emerging research around circulating tumor cells, as well as this whole idea of circulating cell-free DNA, that you can just take a blood sample and based on the blood sample identify driver mutations, say, in a patient who's got lung cancer. What are your thoughts on that? Are you all involved in any kind of research dealing with some of these more futuristic testing methodologies?
So I think it's a fascinating concept, and a lot of that technology was worked out in trying to associate circulating tumor cells with prognosis. So I think at least the initial platforms to do some of that is present, but I'm sure that it's going to take more technology to make sure that we're identifying what we're looking for, not expanding cell populations or doing PCR type evaluations where we might be expanding numbers of targets that are of no significance. So yes, it's a fantastic idea, and if we could count on tumors shedding cells and them being in the circulation and being able to pick them up uniquely, I would be interested in that. I think that's a lot further off than just improving our techniques of biopsy, cell preservation, etc.
Sure. And another question that's come in, just going back to the whole clinical research in the community setting, it seems like a lot of patients come in not necessarily thinking about research, because perhaps then they would have gone to the academic medical center instead. Has your center developed either approaches to overcome some of the patient misconceptions or to provide more education to the patient so that they have a better understanding of what clinical research is and why they would want to participate in it as a cancer patient?
I think if we have a surgical patient, so we also have a biospecimen repository that we collect tissue and store here. So essentially, when the patient's diagnosed with a solid tumor, they're approached for that very straightforward research protocol of discard tissue. And they have an expectation, I think, from that time forward that they'll have an opportunity to participate in research. The medical oncologists here, the radiation oncologists are all very interested in it. We participate in pharma, in device, and in cooperative group trials. So it's just part of the current of what goes on here, and we advertise it too, if you will. It's in our marketing materials.
It sounds like your organization and sort of that encatchment area that you're in, it's ingrained in the culture. It's part of that dialogue. All the clinicians are sort of on board and engaged in it. We found that with some of these other hospitals, as we went out and performed these learning labs, that some that have a very close relationship with a research center, the oncologists were much more attuned to thinking of clinical trial opportunities, some of these smaller hospitals, perhaps more distantly connected with academic centers, it just seemed like it was really, really difficult to even suggest clinical trials to patients because they would have to travel so far and so on. So it definitely sounds very, very interesting. I just want to remind those on the webinar that you can find on the ACCC's website under Provider Resources, there's the Ongoing Advances and Improvements in Molecular Testing. This is a resource that's available electronically. It's also in the July-August issue of Oncology Issues magazine. So I do want to just remind everyone that that is available, and there's a lot more information on the ACCC website regarding these learning labs, some of the lessons learned, and the improvement plans that were developed related to some of these specific barriers that we've discussed today. I'm just wondering if there, as a reminder, if there are any other questions, please type them in. And I guess just one of the other questions that I know that came in earlier was, Dr. Wagman, what are your thoughts regarding this whole awareness now in the community, especially with the president announcing this precision medicine initiative and potentially more patients and their family members hearing about it? Has that changed the tone of either the discussions or the treatment options that patients are even seeking? You know, how has that made an impact in your particular hospital?
I think the overall concept of precision medicine is not just about treatment. It's also about diagnosis and doing the right test at the right time and the right patient. That's part of precision medicine too. And we've been really talking about that at the Cancer Center for at least 7 or 8 years. So that sort of leads off the precision medicine. I also think that the precision medicine that's presented to the public is a little bit misleading. People think that everybody gets a different treatment, and that's often difficult for us as clinicians to explain, because we know how to treat people not because we treat we treat our patients one at a time, but we do large clinical trials that establish the basis for treatment. Now, some person may respond better to one drug or another, or they won't respond at all. So that's sort of very, it's still very gross terms of precision medicine exists now. I think it's going to progress more, and there'll be more precision in medicine, a better operation, a better delivery of radiation, a better drug or drug combination. But I think it's important to still remember that the precision is still based on large populations and their information. So to me, that's an important message to get to the community. And I don't know if I answered the question.
Yeah, no, absolutely. I think it's a great point, and certainly what we're finding also is that every community, in terms of the patient population, their level of expectation and awareness on these kinds of issues really does vary, and it really does depend on how things are communicated to them and the kind of educational materials that they receive. Well, I want to thank you, Dr. Wagman, for your great insights and for your participation today. And at this point, I will hand it back off to Daryl to wrap up the webinar.
Thank you very much, Dr. Kim. And thank you to Dr. Wagman for both of you for presenting today. And of course, thank you to all of you guys out there in virtual land that joined us this afternoon for the webinar. Again, I want to remind you that there's going to be an email sent to all registrants within the next couple of business days. And in that email, you're going to see where you can access a link to access the archive of this recording of this webinar. And there's also going to be a link to the slide deck.